All right. In this particular video, I want to talk about the basically the structure of cardiac muscle tissue and how it's organized. And that basically is this here is the term for how we would describe the layout of cardiac muscle tissue, the cardiac syncytium. And also this video is going to be the beginning of the series of videos I want to make on the physiology of the heart itself. So this would obviously be the first one you would watch as you're going through this. So I'm going to talk about this, and then I'm going to move on to the EKG and then cardiac cycle and so on. So now this particular website that you see right here um, is a website that is an interactive physiology website. That's also the title of it, interactive physiology, that basically allows you to take what I'm going to be talking about with you here and go through this process step by step by step, except with actual interactive videos. So you can click on channels and watch ions move, watch the graphs live and all that. So, go here when you're done watching this video and check this out. So listen to me explain all this to you and then actually watch it in action. This is a really, really good visual tool. In fact, I like to honestly use this when I lecture myself. This site is really well done. And I'm also going to post this under the kind of as a caption underneath the video as well, so you can kind of copy and paste this into the UR into the browser and then go directly there. So just bear in mind when you're done with this video, I strongly recommend you go to this website and check it out. All right. So on that note, let's get rolling. So the first thing I want to talk about in this video is just some basic terminology related to the cardiac syncytium. And then we will talk about the types of cells and then basically how these cells are organized, and then get into some basic electrophysiology of the heart. Basically what I'm saying here is we're going to discuss the action potentials of the types of cells of the heart. And this is very important for you to understand. I mean, whether you're, especially for nursing and medical students, because a lot of medications that you're going to be issuing out to patients revolve a lot around those two processes. So I'm going to talk about just the basics of how the types of cells in the heart depolarize and repolarize. And then later on, when I talk more about like autonomic effects on these tissues and cells, we can get into a lot more clinical or practical application to these with some more specific examples. But for now, I just want you to have a basic working understanding of um, how these cells depolarize and repolarize, because they are a little different than what you have learned in the past about, for example, the nervous system action potentials. All right, so that's how this is going to go. So terminology. The cardiac syncytium is basically, as I mentioned at the beginning of the video, is a word to describe how the tissue of the heart is organized. And the word syncytium means together. And we use this term because even though the heart is like any other organ, and it has, millions upon millions upon millions and billions of cells located inside of it. Except what makes the heart unique is that all of the cells are essentially interconnected with one another. All right, so every single cell is connected to one another. So even though the heart is made up of all all these individual cells, it behaves like it's one unit. So basically, when one of these cells depolarizes, that action potential is transferred into all the other cells that are connected together. So basically, throughout every single cell of the heart. So like I said, the cells have their own individual activities, depolarization, repolarization. But like I said, since all of the cells are going to partake in the action potential, not all at once, but they are going to do so, the heart behaves like it's basically one giant cell instead of all the billions of cells it is composed of in the first place. All right. Now that's an important concept to understand, because in the next video when I get into the EKG, you'll have to understand. So, you've heard me say in the past when I was in my anatomy videos, if we kind of draw a basic layout of the heart here real quick, you've heard me say that the atria do not contract at the same time that the ventricles contract. All right. So basically, the rate of this depolarization is going to vary depending on which cells depolarize first, which ones depolarize more rapidly than others, and so on. All right, but for now, like I said, my goal in this video is for you guys just to have a good basic working knowledge of just how these cells, just the organization of the heart, and the basic mechanics of how these cells depolarize. We'll get more into the rates, like I said, in another video. All right. And something else that you have to understand is basically this term intercalated discs. You'll see in images of this in a second. These intercalated discs are basically these protein structures that you find in between the muscle cells of the heart. So a cardiac myocyte looks something like this. This is a very arbitrary kind of drawing of this. And remember, you just heard me say a second ago that all of the cardiac muscle cells are linked together. All right, and then there are these kind of these protein channels that are found in between the cardiac muscle cells with a few different with a couple different proteins of significance that are found in there. And the two ones that we'll focus on are called desmosomes and gap junctions. All right, and I'll talk more about these in depth in a second. And again, I'll show you these. And then the term striation, by now you better know what the word striation means because you've gone through histology, you've gone through skeletal muscle and how skeletal muscle tissue basically functions and the physiology of all that, and you know that striations are stripes, all right? So cardiac muscle cells also have striations just like skeletal muscle, and the reason why is because they have sarcomeres. And these sarcomeres are practically identical to that of skeletal muscle. So basically, in terms of the contractile proteins and the contractile mechanics of a cardiac muscle cell, they're almost identical to that of a skeletal muscle. But the biggest difference lies in how these cells are depolarized and the rates at which they're depolarized. That's very different between a skeletal muscle and a cardiac muscle, and we'll talk about that later on when we get more into the action potentials itself. All right. Um, so there are essentially two types of cardiac cells found within the syncytium. And the two types are the cardiac myocytes, or if we wanted to be more general about our categorization of these, we would call these the contractile cells. And as usual, my pen is going bonkers on me. So we would call the cardiac myocytes the contractile cells, and then the other cells we would call the autorhythmic cells. And there are two kind of subcategories of these autorhythmic cells, which are called pacemaker cells and then specialized fibers. But for now, for the sake of this video, I'm going to describe these both as just, I'm just going to focus on the general aspect of this because they depolarize and repolarize pretty much the same. It's just that, again, what varies realistically between the two is where they're found and the rates. All right, these pacemaker cells are found more in the nodes of the heart, and then in the cardiac conduction system, and then the specialized fibers are found in, well, the actual fibers of the cardiac conduction system, like for example Purkinje fibers. All right, and again, we'll talk more about these later on. But again, how they depolarize is pretty similar. It's just that, like I said, it's really revolves around the timing and how fast they depolarize. All right. So, so again, when I drew this picture here for, a second ago, this kind of square looking cell here, this would be an example of an autorhythmic cell, and then this would be the contractile cell or the cardiac, the cardiac myocytes or the muscle cells themselves. All right, so these are the two cell types. Now let's actually take a look at these all put together. So you remember from histology, from taking a look at cardiac muscle cells, that the organization of cardiac muscle is a little different as compared to skeletal muscle, because remember, skeletal muscle cells are pretty tightly packed. I mean, there's a little bit of space in between. There's a little bit of space in between skeletal muscle cells. And remember, they have these striations and all that. And remember, the space is filled with the connective tissue, so blood vessels can get in here and nerves can get into the muscle cells and all that. But that's one very important structural difference. Um, And then remember, skeletal muscle cells are multinucleated. The nuclei, under a microscope, they typically look like they're on the outside of the cell, but they're really not. And they're just pushed right up against the plasma membrane because of all the contractile proteins inside of the cells. Um, Whereas cardiac muscle cells are, they kind of take on, I don't even know how the world you would describe this shape. But you can get the picture here that they look a little different. So they look a little different. They're shorter. They're branched cells. I mean, these would be the branches. They are striated because they contain sarcomeres, actin, and myosin. Thin and thick fibers. And remember that cardiac muscle cells also are uninucleated, or they only have one nucleus. So that's another big difference between them. All right. Now, the branches, oh, and then also one thing I forgot to mention, there's a lot more space in between cardiac muscle cells than skeletal muscle cells as well. So a lot more space. Um, all right. And now this is another big difference here are these right here. See this kind of white looking line right there? That is an intercalated disc right there. All right, and then remember I said there are two proteins that are found in here. I mean, two major proteins that we're going to focus on that are found in here called desmosomes and intercalated discs. All right, and um, and basically, where these branches are found, all right, that's where you find the intercalated discs joining the cardiac muscle cells together. All right, hence the syncytium. So there are these discs are either going to be joining cardiac muscle cells or these autorhythmic cells to one another. All right, and so like I said, gap junctions and desmosomes are found in there. Okay, so again, you should have a good working knowledge by now of the organization of this, but bear in mind that every single cell within the heart is linked together in this, by this design. All right, so all these muscle cells are linked together with these discs, and these autorhythmic cells are all linked together. And like I said, which allows for that action potential to propagate through every single cell of the heart. And that's kind of a cool design because, like I said, that allows for a certain rhythm of the heart. All right, because remember, the heart is an autorhythmic organ, meaning that it paces itself. Remember, auto means self, so it has its own rate of generating action potentials, and it contracts. And as a result of that, it contracts with a certain rhythm: atria, ventricles, relaxation, and then we start over again. I mean, that's a very simplistic way to think about it, but for now, that's what you need. All right. So again, understand this basic design. And if we kind of break down the intercalated discs a little more, we can first take a look at these desmosomes. Basically, I like to think of these desmosomes as anchors. All right, I have to use the mouse here because my stylus pen is going nuts. All right, like I said, I like to think of these as the anchors of the heart. All right. So these desmosomes, so you can see here how they're linked. Basically, these desmosomes are kind of they're attached to these kind of proteins, these keratin fibers. You can see here within each cardiac muscle cell, and then these desmosomes basically link together with one another within this space you see right here would basically be I'm going to abbreviate this, the intercalated disc. All right. And now these are important because these cardiac muscle cells, they're contracting with a lot of force. And remember, we're saying that when that action potential reaches certain areas of the heart, all these cells are going to be contracting at the same time as a singular unit. So individual cells contract all these cells contract together as one unit. When these muscle cells contract, they're generating a lot of force. You have to think about the ventricles for a second. So let's kind of redraw our heart here again, and pardon my horrible art. Um, Now you have to remember that the ventricles especially have to contract with a lot of force because they're pumping blood out of the heart and basically the right side of the heart member up to the lungs. So that's relatively low resistance. But the left side of the heart, the left ventricles, they have to, the left ventricle has to pump blood out to the entire body through billions of capillaries, found, from head to toe. All right, so that's a lot of resistance. to blood flow, trying to circulate blood through all these billions of microscopic blood vessels in the body, and to work against gravity to get that blood back up to the heart in the first place. So these muscle cells, they're going to be pumping with a lot of force when they're stimulated to contract. So basically these desmosomes help anchor the muscle cells together and prevent them from pulling apart, with all this force that's going on from the constant shortening and then re-lengthening, and all the force that's found in that process. All right, so that's how I like to think of desmosomes. They're basically anchoring proteins in the cardiac syncytium. And then gap junctions are basically ion channels. All right. So I like to think of gap junctions as ion channels. So let's kind of draw again our arbitrary syncytium here. All right. So these pacemaker cells or these autorhythmic cells, when they depolarize, these are the cells that essentially stimulate the cardiac muscle cells to contract. And you understand how depolarization, repolarization works. It's just an influx of sodium ions, an efflux of potassium ions. Now, the heart is unique because calcium is also involved in the action potential as well. And we'll talk about that later on more in depth. But the importance of gap junctions, if we want all these cells to behave as one unit, and we want this, and we want these cells to be contracting rapidly, we have to be able to transfer this ionic current across the syncytium rather quickly. And that's where gap junctions fall in the line here. So gap junctions open and close during certain times of the contraction. So basically what gap junctions do is they propagate that action potential from cell to cell to cell to cell throughout the syncytium. All right. So ions, which basically are the current, transfer are transferred from cell to cell through gap junctions. And then remember that like any other cell of the body, these have a negative internal membrane potential. And cardiac muscle cells typically sit at a resting membrane potential of negative 90 millivolts. All right. So this current so once these positive ions flow from one cell to the next, that's going to disrupt this membrane potential, and then we will start the events of an action potential, which we'll go into more in depth in a second. But that's basically all gap junctions are. Like I said, they're just ion channels, which you can kind of see here. Again, that just transfer the action potential from cell to cell to cell. So when one cell is depolarized, all the adjacent cells are going to depolarize along with it. All right. So again, this is a major contributor into the heart as being, composed of many, many cells, but it behaves like it's one cell. All right. So every single cell in the heart is linked together. All right. So again, when one area depolarizes, the entire heart's going to depolarize as well. Okay. So let's talk about these cardiac muscle cells and the ion channels that they have. So for starters, they've got, they're the typical ion channels. You know, so we'll say black is going to represent sodium. All right. Black is going to represent sodium. We'll say green is going to represent potassium. And what's a little unique here is that there are also calcium channels in here. Okay, calcium channels, and these are the channels that are found in neurons. These are voltage sensitive channels or gates, and these open and close based on electrical changes within the cell itself. And you know that sodium channels are only permeable to sodium, potassium channels are only permeable to potassium, and you can do the math with calcium. All right. And then they just open and close at certain times, depending on which event is occurring during the action potential. All right. Um, and in terms of membrane potential, this is exactly the same as, the concepts are exactly the same as with any other cell in the body. There's a lot of sodium in the extracellular fluid, of the cells, and there's not a whole lot of sodium on the inside. All right, and then same thing with, ooh, should have probably done that in black, my apologies. And then with potassium, there's a lot of potassium inside cells. There's not a whole lot of potassium in the extracellular fluids. All right, and then with calcium, all right, with calcium, there is, there's not a whole lot of calcium on the inside, and there's a little bit of calcium on the outside, but again, just enough to, make the extracellular concentration higher than the intracellular. All right, no, it's essentially, we're talking free calcium ions here, because you have to remember that cardiac muscle cells are similar to skeletal muscle cells in the fact that they have an SR. All right, and remember the SR stands for sarcoplasmic reticulum, and you know that this is basically an intracellular storage facility for calcium ions, which play a pivotal role in the contraction of a muscle cell. Because remember, um, the release of calcium from the SR stimulates, the regulatory proteins of a sarcomere, troponin and tropomyosin, to kind of undergo a conformational change and expose the active binding sites on the actin filaments, so myosin can interact with it. Now, what's a little bit different, this is where one of the differences are found between skeletal and cardiac muscle, is that the sarcoplasmic reticulum is just not as extensive and well developed as as a as a skeletal muscle. So the importance of calcium here is that when we have an influx of calcium, that's kind of the stimulus for the SR to unload its stored calcium to diffuse down into the sarcomere so we can undergo the contractile processes. All right, that's the importance of the influx of calcium here within a cardiac muscle cell. Whereas with skeletal muscle, the SR is more well developed, it's more extensive. When that wave of action potential propagates down the T tubules to the sarcoplasmic reticulum, that know that there's going to be plenty of calcium release that's going to get down into the sarcomeres, and then we can have the the cross-bridge cycling after the calcium is released and the preparation phase is complete, which I like to call, well, which is called the excitation contraction coupling. All right. Um, so that's the importance of calcium here. And one thing I'm going to mention right now, I'm going to bring it up again. The stronger the influx of calcium into the cell, the more force there's going to be. So basically what I'm saying here is the more the calcium that diffuses into the cell, the stronger the contraction. All right, that's important to keep in mind. All right, and especially if you're thinking about medications like calcium channel blockers, or medications like digitalis and so on. All right, those play an important role at these calcium channels and how much force the heart is going to contract with in the first place. All right, so that's something you have to keep in mind. And then these autorhythmic cells, basically similar in design. They've got sodium channels, they've got potassium channels, and they've got some calcium channels in them. All right. And again, just basically the variability between these autorhythmic cells is how highly concentrated they are with these sodium, potassium, and calcium channels, especially sodium channels, and how quickly they open in the first place. All right. So for example, in the AV node, all right, these calcium channels do not open as quickly, which delays the movement of the current from the atria to the ventricles. But that's a good thing, all right, because we've talked about this basic concept before, the importance of the atria contracting and then the ventricles remaining relaxed. All right, so when the atria are contracting, all right, the ventricles are relaxed and filling with blood. And by delaying that action potential from moving from the atria down into the ventricles, that allows for just a little bit more time for the ventricles to fill with blood. Because the more blood the ventricles fill with, the more blood that can be pumped out of themselves. All right. So that's something to keep in mind. So these autorhythmic cells are designed, like I said, even though they look different and they behave differently, they have the same, they have essentially the same um, channels that a cardiac muscle cell would have. So then if we take a look at the cardiac syncytium in general, um, again, we would see these sodium channels, potassium channels, and a little bit of calcium. Okay. And then same thing with the cardiac muscle cells, sodium, calcium, and potassium. Okay. So and then remember, in between in between all the cells, there are those intercalated discs. All right, those intercalated discs that where you find those desmosomes and gap junctions that allow for that action potential to be propagated from cell to cell to cell. Remember when I say the word propagate, you just think the action potential is moving or being transferred from one area to the next. All right, if we're talking about a neuron, if we say that action potential propagates down the neuron, it's just that current is just working its way down the length of the axon itself. When I'm saying that in reference to the cardiac syncytium, that action potential is moving from cell to cell to cell. Okay. Now, these autorhythmic cells are basically the cells that are ultimately going to be stimulating the cardiac muscle cells to be depolarizing. All right, so these have very oscillating membrane potentials that they have very oscillating membrane potentials that are kind of constantly spontaneously depolarizing, repolarizing, depolarizing, repolarizing, depolarizing, repolarizing. And basically, as these depolarize and repolarize at certain rates, remember that once the current, once the depolarization occurs, it's going to be transferred to the adjacent cardiac muscle cells. And then this cell is going to depolarize, and then the current or action potential will be transferred to this cell. Then this will undergo the events of depolarization, repolarization, and so on. You get the picture. All right. Um, so that's something to keep in mind. Now, what I want to talk about next is basically how cardiac muscle cells depolarize. Now, remember, as you're taking a look at this for a second, now you're thinking you're traditionally taught, when there's depolarization, repolarization, there's this woo, up spike, down spike, and then boom, and so on. All right, this is basically what nervous tissue would look like. All right, because the rate of depolarization and the whole action events of an action potential in a nerve are going to be 3 to 5 milliseconds at most. All right, when we're talking when we're talking a cardiac muscle cell, we're talking upwards of 300 milliseconds, 300 to 400 milliseconds. All right. Now it's important that it takes a little longer for depolarization to occur, and I'll talk about why in a second. And we'll also talk about the events that occur. So there are basically four phases that go into a cardiac myocyte action potential. Four phases. All right, I'm going to put the numbers on here right now. So four, 0. That's supposed to be 0. My apologies. 1, 2, and 3. Okay. Okay. So let's talk about this process. So when a cardiac muscle cell, when a cardiac myocyte is stimulated, when it is going to undergo the events of an action potential, um, like I said, there are four events that occur, and you can see that this graph looks significantly different than that of a neuron. All right, because typically, I mean, it's just depolarization, repolarization, a little bit of hyperpolarization, and then boom, we're back to normal. Okay. Now, let's talk about these phases. Let's talk about these phases here. So phase 4 would be when the muscle cell is at, I'm going to abbreviate this, resting membrane potential. Now, let me erase this real quick. All right, resting membrane potential. So I should put RMP. Okay. And remember that the resting membrane potential of a cardiac muscle cell is negative 90 millivolts. It's supposed to be an M, but my computer's having a seizure on me here. My apologies. So negative 90 millivolts. Okay. And then remember that these autorhythmic cells are constantly depolarizing, repolarizing all the time, and transferring that action potential to these cardiac muscle cells. And basically, when that wave of depolarization works its way into each cardiac muscle cell, we're going to undergo these particular phases. So while the muscle cell is at rest, it is in phase 4. All right, phase 4. So basically, it is at negative 90 millivolts. Now, once that wave of depolarization is transferred or propagated into the cardiac muscle cells, what's going to happen then is you're going to see an upward spike. Okay. And basically, this upward spike is just like with the regular action potential with the neuron, which you already know by now is an influx of sodium ions, okay? And there are these very fast channels that open, meaning they open really quickly and they allow for a rapid influx of sodium into the cell. Okay, a rapid influx of sodium into the cell. All right. And basically, you understand depolarization, we're becoming less negative. And so much sodium is going to work its way into the point where we'll go, you know, above 0 and become slightly positive. All right. Usually we don't get much higher than about positive, 25 millivolts. Okay. So then once we reach this very positive charge here, then what's going to happen are you know what's going to happen. The sodium gates are going to close. All right. And you understand what happens next, because you understand how a neuron works. Well, then these voltage sensitive potassium gates are going to open. All right. These potassium gates are going to open. Now, and then due to the highly positive, the very positive charge of the internal environment of the cell, that's going to start to drive potassium out. All right. But this is where it gets a little different. So that's basically what phase 1 would be then. The sodium channels would close and then we start to have an efflux of potassium. Now, once we get back to about 0, what's going to happen is those other channels are going to open, the calcium channels. Okay, calcium gates are going to open. And then this is going to cause an influx of calcium. Okay, and then what this is going to do, because we have basically equal amount of sodium of these positively charged currents moving into and out of the cell, potassium and potassium out, calcium in. There won't be a whole lot of potassium moving out. All right. So then, as calcium works its way in, what's going to happen? Remember, this stimulates the release of calcium from the sarcoplasmic reticulum so we can undergo the contractile phase. Now, if we take a look at this for a second, let's say this blue line is going to represent the force of contraction. All right. So what you notice here is that as the calcium influx occurs, that also means that there will, that the heart is going to contract with more force. All right. And then basically what you see here is basically what phase 3 is. This is when the calcium influx stops, and then you see the events just as regular as you see over here. You see the efflux of potassium out of the cell. All right. And remember, the more, the longer these calcium channels stay open, the more calcium moves into the cell, the greater the force of the contraction, because the more calcium that's released, out of the SR into the sarcomere of cardiac muscle, the stronger the contraction that's going to take place. Okay. The stronger the contraction that's going to take place. Um, See then, and then basically, like I said, phase three is just the movement of potassium out, and then we will return back to resting membrane potential, and then this process will start all over again. And now the big difference here is this plateau phase. This extends the action potential out a lot longer. Instead of being 3 milliseconds, about 300 milliseconds. All right. Now, this is important because the slower that these muscles contract, the more time there is for filling. All right. The more time. This is especially important with the ventricles. All right. So if these cardiac muscle cells contract, if it takes a little more time to actually get these cells contracting at full force and propagating that potential to the next cell, that means that the ventricles or I mean the chambers themselves, but this is especially important with the ventricles, are good. There's just going to be more time for the chambers to fill with blood. That's the importance of this plateau phase or the delay in the action potential. Okay. And you understand, and as well, that sodium issues don't really affect the heart all that much. I mean they can if sodium imbalance is very great, but and that potassium problems, whether you're hyper or hypokalemic, are very dangerous to the heart. And then we can also add calcium to this as well. So calcium and potassium electrolyte imbalances can have direct effects on the heart. And we'll talk about this later on in another video, but you have to keep that in mind. So these essentially are the events of an action potential of a cardiac muscle cell. Again, we go from resting membrane potential up, to these fast sodium channels open, and once the muscle tissue is stimulated to contract or depolarized, and then the sodium channels close once we reach a kind of a positive charge of the cell, and then potassium channels open and we have a slight efflux of potassium, and then once we kind of get right back down to zero here, these calcium channels are going to open, which maintain this plateau and keep the cell at a charge of about zero. And then eventually these calcium channels are going to close, and then potassium will more potassium gates will open and more potassium will move out of the cell. And you know how this works. Then we will go back to our resting membrane potential. And then, just like in other cells, there are sodium potassium pumps that reset this ionic balance between the intra and extracellular fluid. That sodium is, 3 sodium ions are pumped out, 2 potassium ions are pumped back in. And then there are also these sodium calcium pumps that are important for moving calcium out of the cells as well, because we have to get that calcium out of there, to return this, because remember, calcium is a positively charged ion as well, so we need to get that out of there, so we can restore um, the charge of negative 90 millivolts. All right. So I hope that wasn't too confusing, because I know I did a lot of doodling and it's kind of random on here. But these are the events of a cardiac action potential. Now let's take a look at pacemaker potentials, or basically the potentials of the autorhythmic cells. Now, what we have here is we have 0 and we've got 3. All right. Phases 1 and 2 are absent in the autorhythmic cells of the heart. All right. They're absent in the autorhythmic cells of the heart. All right. Now, like I mentioned before, these cells have a constant, just this oscillating membrane potential all the time. They're constantly just spontaneously depolarizing, repolarizing, depolarizing, repolarizing. All right. And then as these cells are constantly depolarizing, repolarizing, they're going to obviously Goodness gracious. They're going to transfer that action potential from cell, into the cardiac muscle cells. And then as these cardiac muscle cells are, stimulated to contract, then they will transfer that action potential from, cell to cell to cell. All right. So like I said, what's a little different here is that now notice the upswing, the slight depolarization of the, phase four here is a lot slower. Okay, it's a lot slower. And that's because of the presence of these sodium channels, which are called IF channels. The F actually is kind of cool, stands for funny. All right, because these behave a little differently than the fast sodium channels that I was talking about. All right. Now, the you know we could say that about the resting membrane potential of the autorhythmic cells of the heart is about negative 60 millivolts. All right. Negative 60 millivolts. Now, being that these autorhythmic cells typically don't drop down to a resting membrane potential of negative 90 millivolts. All right, that prevents these fast sodium channels from opening at all. All right, so in a regular autorhythmic cell, the fast sodium channels are not involved. All right, so these slower, funny sodium channels, these are the ones that are normally operative here in the cell. So what happens, and we call these slow sodium channels because they open slowly and they cause a much slower influx of sodium into the autorhythmic cells. All right. Now, eventually, once the autorhythmic cell depolarizes to about negative 40 millivolts, all right, then what's going to happen are calcium channels are going to open. All right, calcium channels are going to open. All right, and these are a little goofy as well because these calcium channels are a lot faster than that of the cardiac myocyte here. So with this plateau phase, the calcium is moving in relatively slowly. With the, with the basically with the massive upswing and the depolarization um, of what was I going to say? Of these autorhythmic cells, calcium moves in a lot more quickly. All right, and then, excuse me, and then basically will depolarize, a little bit beyond zero as well. And when that occurs, then, well, the events are going to be the same. As kind of these, this is going to be a lot more similar to that of, I should say, of a neuron. All right, so then potassium channels are going to open, and then potassium will move out. All right, so obviously once we kind of get to this threshold for the inward movement of calcium, then these calcium channels close, potassium opens, and then you know, potassium goes out. And then again, just, like with any other cell, we've got the sodium potassium pumps, moving potassium and sodium out. And then we've also got those calcium pumps, all right, moving calcium and, kind of sodium out, exchanging them as well. All right. Um, what was I going to say here? Shoot, I lost my train of thought. Good. Oh, and then once we repolarize back down to about negative 60 millivolts, then immediately that's going to cause these slow sodium channels to start opening again, and then we just start this process all over. And then that's why I said that these autorhythmic cells have kind of an oscillating membrane potential. So the second they get back down to negative 60 millivolts, the sodium channels are opening again, and this process is just starting all over. So, like I said, these autorhythmic cells just kind of have this inherent rate at which they're depolarizing and repolarizing at. All right. And some of these cells depolarize and repolarize at faster rates than others. All right. For example, if we think about the cells of the parts of the cardiac conduction system, there's an SA node, there's an AV node. All right. There are bundle branches. and in between here, I forgot the bundle of His, sorry. And then there are these fibers, I'm just going to abbreviate this, called Purkinje fibers. All right. All of these are components of the cardiac conduction system that have these autorhythmic cells that depolarize, repolarize at certain rates. Now, the cells within the SA node have the highest rate of depolarization, repolarization. That's why the SA node is called the pacemaker of the heart. All right, because like I said, the cardiac syncytium is basically many cells joined together as one unit. All right, so whichever cells depolarize and repolarize the fastest, all right, that means that action potential is going to spread through the heart from the quickest area of the heart, and that's the SA node. So then basically all these other structures of the conduction system fall in line, basically depolarize and repolarize at this rate, because the action potential is moving, through all these structures because of the speed of the SA node. And that's a process called overdrive suppression. Basically the speed of the SA node of the heart, all right, kind of suppresses the normal oscillating rates of the rest of the conduction system. So then let's say, for example, if a person's SA node fails, which does happen, well then the part of the conduction system that has the autorhythmic cells that depolarize, repolarize the fastest are then going to take over, and then that would be the AV node. All right, so then the AV node would pace the heart, but it would do it at a much slower rate than that of the SA node. Okay, so you get the picture, and I'll talk about the conduction system more in depth later on. But again, the basics of this are important to understand, especially which ions are moving in and out and when, because like I said, when you're dealing with patients and medications that are being given out to patients, um, this is where those medications are taking their effects, is they're playing a role in, well, which of these gates are closing, which of these are opening, are they going to open or close more slowly or whatnot. All right, so you have to keep that in mind. All right, you have to keep that in mind. So, boy, I hope I didn't confuse you too much with this, but in a nutshell, this is basically the cardiac syncytium, and how basically how the tissue and all the cells are organized, like I said, into one, like I said, many cells that behave as one unit. All right, many cells that behave as one unit. And like I said, the cells that are pacing the rate at which the cardiac muscle cells are contracting are the autorhythmic cells of the heart. Okay, so these autorhythmic cells with these oscillating, depolarization, repolarization, depolarization, repolarization rates basically affect the rate at which the cardiac muscle cells are going to contract. Okay, so you have to keep that in mind. And then, like I said, understand the basic events of each phase. Again, phase 4 of a cardiac myocyte is just resting membrane potential. And then phase 0 would be the fast sodium channels opening. And then phase 1 would be the sodium channels closing, potassium starting to move out. But again, then phase 2 would be the plateau phase. Potassium moving out, but an equal amount of calcium also moving in, creating the plateau. And then when the calcium channels close, then all we have is potassium moving out, phase 3, and we return back to our resting membrane potential in phase 4. And then when these autorhythmic cells doink, when they depolarize again, then this process is going to start all over again. All right, and then remember that these autorhythmic cells, that they are lacking phases 1 and 2, just due to just their design and their nature. Like I said, these have a resting membrane potential of about negative 60 millivolts. All right, and again, which suppresses the activity of those fast sodium channels, which are present, they just don't open. All right, because remember, these are electrically sensitive channels, and since these cells, normally don't dip down to negative 90 millivolts, those channels aren't going to open. So these cells depolarize at a slower rate, and then once we reach, I guess in a sense we could call negative 40 millivolts the threshold. all right, the threshold at which the faster aspect of depolarization will occur. And remember that once threshold is reached, calcium channels will open, and then we'll have a massive influx of calcium into these cells. And then once we reach a certain point, a certain charge, calcium channels close, potassium channels open, and then we depolarize back down to negative 60 millivolts. And then immediately when we get back down to negative 60, then this starts all over again. And then, like I said, as I mentioned a little while ago, the clusters of these autorhythmic cells that depolarize and repolarize the fastest are basically going to set the pace of the heart, or they're going to set the heart rate. All right. And like I said, the SA node, which stands for the sinoatrial node of the cardiac conduction system, has the cells that do this the quickest. So as a result, the SA node, that's why it's called the pacemaker of the heart. All right. And so, like I said, I hope this all made sense to you or shed some light to you. If it didn't, or if I just wound up confusing you even more, like I said, check out that website, the interactive physiology website that I posted on the initial slide here, and that I'll put in the caption. Because, like I said, I can sit here and explain this stuff to you all day, but you really have to see it in action to really draw out an appreciation of how this works. So go to that site when you're done with this lecture, or even in the middle of it. Once I get, especially like for example, once I got to, once you get to this and you hear me listen to it, stop and then go to that site and then watch this step by step. It'll make, like I said, it'll just make this make a lot more sense and bring a lot more life to it. And as usual, if you have any questions, don't hesitate to ask me. I like to do what I can to try to get back to people. I know sometimes a little slow, and I apologize for that, but I'm very busy all the time. And just keep working hard, and good luck in your studies.